Good afternoon, everyone. My name is Marlene McKinney, and I'm presenting on Donepezil's role in mitigating opioid-induced respiratory depression without compromising analgesia in B6 mice. So as many people already know, fentanyl is a very potent opioid that stimulates the mu-opioid receptors in the body, which can result in a number of effects due to the sedative impacts of the drug. This results in, for example, the sedation of key respiratory centers in the brainstem and other parts of the nervous system to result in rhythmogenic changes, frequency changes, as well as amplitude changes when it comes to breathing. And it also can impact sleeping, as I will go into in a bit. And these sedative impacts will result in opioid-induced respiratory depression, or OIRD for short, which can cause death and is the #1 cause of death from opioid overdose. And I have this figure at the bottom here that shows the comparison between different states of wakefulness and fentanyl intake at only 100 micrograms per kilogram. And as I said before, fentanyl only fentanyl will sedate sleeping processes. So this is not even during slumber, where the frequency of breathing is at a 38 breaths per minute, which is very dangerous, as you can see from the fact that it's much higher in the sleep states and, of course, the wakefulness states. Acetylcholinesterase inhibitors, such as Donepezil, as I touched on in my presentation title, have shown promise in improving respiratory function in preclinical models of OIRD. And Donepezil is one that has been used in Alzheimer's disease for treatment of that. And the studies of human taking opioids for cancer pain as well, when that's co-administered with acetylcholinesterase inhibitors, has been shown to improve sedation and fatigue. And of course, as in the name of the drugs, they increase the cholinergic neurotransmission to increase wakefulness. But the specific effects of the acetylcholinesterase inhibitors, such as Donepezil, co-administered with fentanyl in particular, remain poorly understood due to lack of studies with that. So we hypothesized in my lab that subcutaneously administered acetylcholinesterase inhibitor Donepezil will attenuate fentanyl-induced respiratory depression without compromising analgesia or lack of pain reception in freely behaving B6 mice. So our subjects were adult B6 mice bred in the Seattle Children's Research Institute vivarium, and we had 3 cage groups for each of our treatments in our within subjects design, which meant that all of our cages would receive one of our treatments, which I'll go into when I'm going through our experimental design. We measured our breathing using whole body plethysmography, which was done in the chamber up here. So the mice would sit in the chamber and have their breathing tracked over time. And our measure of nociception, or that pain tolerance or lack thereof for the pain reception, was done through tail flick latency tests. So the mice would be put into this chamber here, and their tail would be placed through a slot where we would dip it into a hot water bath and time the amount of time that it would take for them to take it out of that hot water. So in our experimental design, as was put on the other slide, we conditioned them to the chambers and our tail flick latency interface for about 5 days before experimenting. Then we did the baseline tail flick latency over video for precision. And then we had 1 of 3 injections that would occur with a cage group on a given day. So it'd either be a fentanyl only injection, a co-administration fentanyl plus Donepezil injection, or our vehicle control injection of saline. And then we would put them in the chamber to have them record the breathing for an hour. And then we would do our post-drug tail flick latency after that and take the video for that for precision as well. So our anti-nociception results were studied using 2-tailed t-tests. And we found, as expected with our saline, since it was our control, that there was no significant increase or decrease in our saline tail flick latency between the pre and post-drug values. With our fentanyl, there was a 34% increase of our TFL values, and with the co-administrative injection, there was a 56% increase in the timing. However, unfortunately, with our respiratory attenuation results, there was not significance with them. We calculated the percent maximum possible effect and utilized repeated measures one-way ANOVA with Bonferroni's post-hoc test in order to adjust for our results. But we found that we perhaps used a too low of a dosage of Donepezil in trying to bring back the respiratory activity that they should have normally. To conclude, the dosage of that Donepezil that we gave them, which was 0.5 milligrams per kilogram of Donepezil, co-administered with 0.1 milligrams per kilogram of fentanyl, did not significantly attenuate respiratory depression. However, Donepezil might have enhanced the anti-nociceptive properties of fentanyl, as we saw that difference in the increase with the tail flick latency times of 56 compared to 34% with just the fentanyl alone. However, since our maximum possible effect percentage was not significant, that may have just been due to chance. And we saw with behavioral observations of the mice while they were in our WBP chambers, we saw a return to normal behavior, such as sleeping activity, since we would do it during times when they are usually asleep, as our mice are nocturnal, and going back to normal defecation and urination behaviors. And future studies, of course, can investigate brainstem activity with the co-administration compared to fentanyl intake, as well as using dose response curves to do experiments with varying dosages of both Donepezil and fentanyl to see the impacts of that on breathing. And I wanted to say thank you, particularly to my amazing lab mentor, Zach Lovac, my lab partner, Gabrielle Leon, and of course to my lab PI, Nino Ramirez, and to everybody who made this program possible, including Julie, Sylvia, Dr. Ellen Bogan, and all of our donors, as well as Seattle Children's Research Institute's Summer Scholars Program.